Hello my dear students, I hope you all are fit and healthy. So students, you know that we have started the revision of second chapter, that is reproduction in plants. And students, we have completed the various means by which the plants may reproduce. Number one is asexual, means in that we have studied how the plants may develop by fission method, by budding and by fragmentation and spore formation. Then, my dear students, we have also studied the natural methods and artificial methods of vegetative propagation. And today we will study the tissue culture method. What is tissue culture? It is in vitro aseptic culture of cells, tissues, organs or whole plant under controlled nutritional and environmental conditions. Means we may develop large number of plants using tissue culture technique in a short time. But what are the conditions? That is, we have to develop the plant under aseptic conditions. Aseptic condition means the condition or the atmosphere present must be free of germs. And here the plants that will be developed in vitro. In vitro means under lab conditions. In a lab we will develop new plant. So it is in vitro aseptic culture of cells, tissues, organs or whole plant under nutritional and environmental conditions. We will grow and develop new plant using cell or tissue, right? But under aseptic conditions means condition should be free from germs and the new plant is developed in vitro. Means in a lab we will develop the new plant. So before studying this tissue culture technique in detail, you must know some terms. First one is totipotency. What is this totipotency means? It is the ability of the cell to grow into a whole organism, that is referred to as totipotency. So what is totipotency? It is the ability of the cell to grow into a whole organism. Then the second term you should know, callus. What is callus? It is undifferentiated mass of cells, right? That is known as callus, means group or mass of cells which is undifferentiated. Undifferentiated means it has not attained a specific function, it has not attained a specific role yet. So it is undifferentiated mass of cells, that is known as callus. Then the medium. Medium means we will develop the new plant using some medium, and that in the medium there must be presence of enzymes, hormones and control environment. Then is differentiation. Differentiation means when the callus recognizes its role, then it is known as differentiation. Means the callus we have studied now, it is undifferentiated mass of cell. So the cells here, they do not have attained any role. So differentiation means when the callus it attains its specific role, that is it will develop into root and shoot, that is known as differentiation. So let us study what is this tissue culture technique here. In tissue culture technique, a small piece of tissue called explant is cut off from growing tip of plant. So the term you should know here, that explant. We will take explant. Now what do you mean by this explant? It is a small part of plant. It may be small part of plant. So that is referred to as explant. So from where we are taking this explant, it is cut off from the growing tip of the plant. Why we are taking this explant, that is small part of the plant, from the growing tip of plant? Because at the growing tip of plant we have studied in 7th class that there is the presence of meristematic cells. So meristematic cells are present at the growing tip of plant. The role of this meristematic cell is that they are continuously dividing. So that is why we will take this explant. Explant means a small piece of plant or a small piece of tissue that we will take from the growing tip of the plant, and then what we will do, it is kept in nutrient medium under aseptic condition. We have taken the explant, then we will transfer this explant, that is small piece of tissue, into nutrient medium, right? We have taken an explant, then I am repeating, we have taken an explant, then we will transfer this explant into nutrient medium, right? Into nutrient medium, but the condition must be here that it should be done under aseptic condition. Under aseptic condition means the whole process, that is process of transferring this explant into nutrient medium, must be done under aseptic conditions. Means it should be, the atmosphere there should be free from germs. Then its cells divide rapidly and form a mass of cell that is called callus. When this explant, it is transferred to the nutrient medium, then in the nutrient medium, this small piece of tissue, that is explant, it will grow and develop and it will form callus, right? Okay, what is callus? We have studied that it is undifferentiated mass of cells, right? Then this callus is formed. The callus is transferred to another nutrient medium containing plant hormones. Then this callus, it is again transferred to the another nutrient medium. And in this nutrient medium, specific hormones such as vitamin A is also present, auxin is also present, and cytokinin is also present. Some hormones are also present. What is the need of this nutrient medium? It helps in the growth and development of plants, such as these auxins and cytokinins. They are growth regulators of plant. They help in the development of root and shoot of the plant. So they are basically the growth regulators. Then when this callus is transferred into the another nutrient medium in which specific hormones, vitamin, amino acids, auxins, cytokinins are present, then what will happen? This callus will differentiate. Now we have already studied now what is differentiation. Means when the callus is attaining a specific role. Now this callus will divide and it will form root and shoot, and then the root and shoot will develop, and finally it will develop into a plantlet. So let us see here, the callus is transferred to another nutrient medium containing plant hormones. The cells of callus in this nutrient medium divide and differentiate into root and shoot and form several new plantlets, and new plantlets will be formed. And these plantlets then will be transferred to the pot which is filled with soil, and that will develop into a plant. So this is the way how we can develop a new plant using only a small piece of tissue from a plant. So this is a tissue culture technique, and it is also referred to as micropropagation. So what we have studied in tissue culture, here we may develop a new plant under aseptic conditions, right? Using only a small piece of tissue. So tissue culture is also known as micropropagation. Now we will study the next process, that is sexual method of development of plant. We have already studied the differences between asexual and sexual means of development of plant. Let us study this process in detail now. Here we know two parents are needed and there is the need of gametes. So it takes place by the formation of gametes, and two parents are needed in this process. Now before studying this process in detail, we must know the flower, that is the reproductive part of the plant. So we should know its detail. So first one is androecium. What is androecium? It is the male part of plant. So in flower, which is the male part? That is the androecium. This is the androecium. Androecium is also known as stamen. It is formed of two things, anther and filament. The upper portion is known as anther and this is known as filament. So androecium is known as male part of flower and it is formed of two things, that is filament and anther. Then it is containing the male gametes. So male gametes are formed in the androecium. Now we will study the female part, right? So female part of the flower is gynoecium, and gynoecium is containing the female gamete, which is known as egg, right? It is formed in the ovule. So let us study before this what is ovule. We should know what is gynoecium. Gynoecium, it is consisting of stigma, that is the upper portion. Then it is long slender-like structure. It is known as style, right? Then there is a ovary. So stigma, style and ovary, these three things, they are forming the gynoecium. And just as androecium is known as stamen, similarly gynoecium is known as with another term that is known as pistil. So pistil or gynoecium, it is formed of three things, that is stigma, style and ovary. The upper portion is known as stigma, then comes the style, then is the ovary, and inside the ovary there are ovules present, and female gamete, that is egg, is formed in ovule, right? So what we have studied now, that is male and female parts of flower. So male part is the androecium, which is consisting of two things, that is anther, the upper portion, and then is this filament, and they are also known as together as stamen. Then is the female part of flower, that is known as gynoecium. Gynoecium is also referred to as pistil, and it is consisting of three things, that is stigma, style and ovary. The upper portion, it is known as stigma, then is the style, then this is the ovary. And this stigma, it will receive the pollens, right, which will be formed inside the anther. So this anther, in this what are present, pollen grains are present, right? So which will be containing the male gamete, and this male gamete will be received by the stigma of the female part. Then fertilization will occur through the formation of the pollen tube, that we will study in the another video in detail. So let us just study here about androecium and gynoecium. Then there is a pedicel. Pedicel is the stalk of a flower, and the swollen or the terminal part of the pedicel is known as thalamus. Then comes the sepals. Sepals are the green leaf-like parts in a flower, and group of sepals is known as calyx. Then there are brightly colored petals, and group of petals, they are known as corolla. So what we have studied in this flower, we have studied first one is androecium, then we have studied gynoecium, then about pedicel, thalamus, sepals and petals. So this is about flower. So students, this is all for today. Take care of yourself.